Hello and welcome, dear friends, once again to the most fascinating discussion on the DNA, apart from the central dogma process which you have seen, that is replication, transcription, translation. This is an event which takes place when there are abnormalities and anomalies on the DNA molecule. When there is a wrong incorporation of a base, when a base changes its chemical nature, when base analogous compounds are there, when alkylating agents they try to disrupt the normal processes of DNA, the cell it evolutionarily has harbored a mechanism by which it can overcome these damage or the lesions on the DNA. So today I will be discussing two such mechanisms in my lecture over here: nucleotide excision repair, or popularly known as NER, and base excision repair, also known as BER. The third is mismatch repair system, which I would have loved to include in this discussion, but just for avoiding the saturation of the topic or crossing the time limit of a standard lecture, I am keeping this mismatch repair system as a separate lecture. So friends, as you know very well, mutagenic agents, they induce a mutation. Apart from that, spontaneously there are mutations in the DNA, and the cell has to overcome this for its survival in the present generation. So repair is such a beautiful mechanism which evolution has sustained in all the cells, prokaryotes and eukaryotes, that they act with the help of certain set of enzymes. These enzymes they come at the point of lesion, means lesion or a damaged DNA, it invites the enzyme array, and these enzymes are different in different species across the life systems, plants, animals, microorganisms, fungi. All they harbor different variety of enzymes, different quantity of these protein molecules. Some are dimers, trimers, tetramers, protein complexes, and some have a multiple functions in the same protein molecule. Some they require, each step requires a different protein molecule. So across the domain of life, if you try to explore these repair mechanisms, you will find a huge variety in this. But the basic steps which are there at this graduate level, you have to understand the basic steps which are common, means recognition of a wrong base, removal of that base, filling it with a correct. This basic principle is same across life. So today we will see two such repair mechanisms: nucleotide excision repair, base excision repair, mismatch repair in my next video. Now the pattern of this lecture I have kept for nucleotide excision repair. You will see a small animation first, animated version of the presentation, followed by a PowerPoint version for you to take down the notes and for felicitating better understanding of the topic. Same with the base excision repair, a small animation followed by the PowerPoint presentation. I hope you understand, comprehend, go to the higher level of understanding, get yourself prepared not only for the postgraduate studies, but I would challenge you to go for research, because hundreds and hundreds of such repair mechanisms are available in nature which are still unexplored. In 2015, some 6 years back from this day of my presentation, this one of the base excision repair enzymes got a Nobel Prize. Think about it. Uracil DNA glycosylase, it got a Nobel Prize in 2015. So there are huge opportunities in the research also. We don't know how many types of repair mechanisms may be still there unknown to mankind. These 3 to 4 repair systems which we are seeing, let us consider that as the base for you to understand the repair mechanisms, and on this basis you may go for the higher domain of understanding this fascinating molecule which is known as the deoxyribonucleic acid, or the DNA. Come, let's have a journey of knowledge with respect to NER and BER today. So come, let's take an animated version survey of how DNA repair mechanisms it functions when there is a nucleotide excision repair. So there are many microbes in the environment which commonly encounter this ultraviolet light, which can cause the neighboring pyrimidine bases, that is the thymine dimer, to form a kink or a duct, as you can see, a duct, and a nucleotide excision repair is triggered off. All these functions are enzymatic, enzyme based, and whenever we think about an enzyme, there is a specificity with respect to the substrate needed, and this substrate is this damaged DNA. So this damaged DNA induces the enzymes to come and act on this particular type of environment. So pyrimidine dimers, they can be repaired by several mechanisms, and one of the method is nucleotide excision repair. In this system, there are three proteins. There are three proteins which are made up of you can say a product of gene expression, and that is product of Uvr A gene, Uvr B gene, and Uvr C gene. In this three subunit exonuclease protein system, this is, as you can see here, two A molecules, one B molecule and one C molecule. They excise a path of 12 to 13 nucleotide excision to the left and right of this dimer formation. If you recall, in your photolyase enzyme system, there was only the thymine dimer, that is the cyclobutyl ring removal. So there is no loss of DNA there. But in nucleotide excision repair, not only the damaged DNA is removed, but along with it some good bases are also removed. So it excises a path of 12 to 13 nucleotides spanning the dimer. ATP hydrolysis is required throughout the system, and as you can see, 12 to 13 MERs, these are called as 12 to 13 MERs. These 12 to 13 MER DNA is the main point of attraction for these enzymes or these proteins, I can say. So the repair mechanism begins with the association of Uvr A and Uvr B and the hydrolysis of ATP. The Uvr AB complex now binds to the DNA. As Uvr A protein, it makes the initial recognition of the DNA damage. Now here one thing which has to be understood is that this Uvr A, which is bound to the dimer along with Uvr B, at each step ATP hydrolysis is required. This is a high energy requiring process. Now once it is bound here, Uvr A will bend the DNA near the damage. This will be completely bent, and Uvr A will load on Uvr B onto the damaged DNA, after which the Uvr A complex will be removed. This will be ejected from the whole assembly on the DNA. So you can see here the bending is there, again ATP is required, Uvr A is ejected from the complex. Now once this happens, next the Uvr B which is there, it will bind to Uvr C. This Uvr B will bind to Uvr C, and not only that much, the nick will be there. The 13 MER will be removed by another enzyme complex, which is helicase. The single-stranded gap will be filled by DNA polymerase, as it takes place in regular scenes, because the 3' hydroxyl end is here is free, and whereas the gap which is there, that will be sealed by ligase. The single-stranded gap which is resulted due to the removal of this 12 to 13 MER by UVRD, that is a helicase enzyme. That helicase has dual function. It has the capacity to bind on to the DNA, rotate, swivel, and just go on degrading. It has an endonuclease activity as well as an exonuclease activity, and this helicase, it removes the strand, as you just saw here. I will rewind it for your convenience. So once this is over, as you can see, the nick is there. I have rewinded it. So you can see now another complex called as the UVRD helicase. It strips the resulting short single-stranded damaged DNA from the DNA molecule. This is UVRD molecule. The single-stranded gap now will be filled by DNA polymerase here. As you can see, it is just adding. Ligase will seal the nick. Although in this example which I have shown you exclusively is a example of pyrimidine dimer, cells can use this for nucleotide excision repair to fix large number of damages which we have seen in the types of which we have seen in the types of mutations. So come, let's have a more detailed look of repair of damaged DNA by nucleotide excision repair with respect to some PowerPoint slides where you can take down the notes and also have a better insight of what this process is all about. So as you can see, this is one of the type of DNA repair systems, and we are having a microbial perspective or a Escherichia coli perspective. Nucleotide excision repair, the enzyme system which you just saw in the animation, that is which involves all the A, B, C, D proteins and the helicase enzyme, DNA polymerase, ligase enzyme, all this complex is called as a ABC excinuclease complex. So the excision part of that enzyme, which is cutting and removing the bulky lesion, is called as ABC excinuclease. So these DNA lesions which cause large structural changes, that is the pyrimidine dimers, are removed by this ABC excinuclease. So after understanding nucleotide excision repair, you should have a vision where you will understand what is nucleotide excision repair, how does it differ from the photolyase enzyme system, how it is different from base excision repair and the mismatch excision repair, which you are going to see in the succeeding lectures. So nucleotide excision repair is different in prokaryotes and eukaryotes, and if possible you should be able to draw a line of difference between that. This is for your PG level studies, it will be helpful, and those who are applying for competitive examinations should not neglect this, even though if I am not telling here and it is not present in your syllabus. But nucleotide excision repair involves a much more complex and a larger quantity of proteins and enzymes in eukaryotes as compared to the simple 4 to 5 proteins in prokaryotes. Next is enzymes involved in the prokaryotic nucleotide excision repair. Each and every enzyme and its function you should be able to describe, and the molecular mechanisms of nucleotide excision repair. These are the things which you should be able to know, comprehend and able to reproduce when you will be studying nucleotide excision repair in prokaryotes. So let's have a detailed look of what is this nucleotide excision repair all about. This is a type of DNA repair mechanism, as you have already now come to know. It can be defined as, the definition of NER can be put forth as a type of DNA damage and a repair mechanism where the damaged base along with a short stretch of healthy strand is removed and later on the gap is filled with correct nucleotides by the polymerase enzyme. This mechanism is a cut and patch mechanism. Nucleotide excision repair, it functions by a cut and patch mechanism. It is present in certain prokaryotes and eukaryotes, but the components and the mechanism, it varies to a great degree. The basic fundamental process is same, but I would like to mention here eukaryotes have a more larger number of proteins and they vary with different species, means from animal to animal, organism to organism, from cell to cell, there is a variation in the number and the type of the protein quantity and the mechanism involved. But nevertheless, the 4 to 5 steps which you have seen, that remains the same. Now this nucleotide excision repair can remove a variety of bulky lesions. Bulky lesion is exactly opposite of the small lesions, means a thymine dimer only 2 nucleotides getting adjacently attached is a small lesion, it is a short lesion. But if there are many such lesions on the DNA, it will be called as a bulky lesion. And if you see the photolyase enzyme system, it was involved in removal of small patches only, small lesions. But nucleotide excision repair is capable of removing bulky lesions like the pyrimidine dimers and chemically modified bases. Now nucleotide excision repair has certain enzymes in its machinery, means the prokaryotic enzymes, collectively these are called as the Uvr, that is the repair which is there, ABC endonuclease system. Okay, this is done by the Uvr Abc endonuclease system. This whole complex is called as DNA excinuclease. Endonucleases are those enzymes, I would like to mention here, those which recognizes free ends of the DNA. DNA is basically covalently closed and circular. But if there is a nick in the DNA and some free ends are there, these free ends are immediately recognized by endonuclease enzyme and they are degraded. They are cut forth. So this nucleotide excision repair in prokaryotes is primarily initiated, or the cut is initiated by endonuclease enzyme system, and here it is called as Uvr Abc endonuclease. Uvr endonuclease consists of 4 enzymes: Uvr A, Uvr B, Uvr C, Uvr D. Uvr B has endonuclease activity, so does has Uvr C, and Uvr D it has a helicase enzyme. So these are all the functions same as which you have seen in the DNA replication. So Uvr Abc complex, which is also called as a DNA excinuclease complex, as you saw in the beginning of the animation, this enzyme has the capacity to cleave 2 phosphodiester bonds at the same time. Which are these 2 phosphodiester bonds? One to the left of the kink or the dimer and one to the right of the dimer, encompassing some 12 to 13 nucleotides in the middle. So this NER also requires DNA polymerase and DNA ligase later on when it leaves the whole strand with gaps. The mechanism, if you see in the bacterial system, Uvr Ab complex, it scans the DNA first. This Uvr A and B, it scans the DNA. Now you can find some ambiguity here. In some cases you will find 2 Uvr A molecules, in some you find 2 Uvr B molecules. All are correct. These are the small variations which are there in different types of species of bacteria, genera of bacteria across the domain of bacterial kingdom. Many species, they have 2A molecules, some have 2B molecules. So to encompass all them, this is a symbolic way of telling that 2 Uvr A and 2 Uvr B molecules. So this Uvr AB complex, it scans the DNA, and Uvr C is recruited, means it is recognized and called at the point of action. Then damaged portion is removed by Uvr D, that is the helicase enzyme which we saw in the animation, and DNA polymerase 1, it fills the gap. One notable thing here of DNA polymerase 1, which we will see in the mismatch repair system, is that it also has an endonuclease function, which is not exercised here, but nevertheless we will see it in the future. DNA ligase then seals the nick. So if you see, if you break down whatever you saw in the animation or whatever I just mentioned, the mechanism, let us split it into 10 to 15 steps and understand it in more clear fashion. So there are 4 to 6 steps. Let us say 6 steps. You can club it into 4, elaborate it into 6. So 6 I will say. First is damaged DNA detection. Second is damaged DNA verification. Third is the damage removal. Then the pre-incision complex, where the damage verification is done, damage is removed, and in the post-incision complex, the replacement and filling up of the gap takes place. So if you see the steps of the Uvr Abc Ner pathway, as it is popularly called as Uvr Abc, which is the enzyme, Ner nucleotide excision repair, which is the mechanism, pathway. So we will split it into as many as steps possible for your better understanding and taking down of the notes. So I split it into, see, number 1, 2 Uvr A and 1 Uvr B combines to form a Uvr AB complex. ATP is required here. So this A and B, it form a AB complex. ATP is required. Now this AB complex has to bind onto the damaged site. That again requires ATP. So Uvr AB complex, it binds and scans for the DNA lesion. Third point, when Uvr AB finds a DNA lesion, it binds there firmly. It remains attached. It halts there. It binds, moves along the DNA, and when it finds a lesion, it halts there. Okay, then 2 Uvr A molecule, they detach off. You can see here 2 Uvr A molecule, they detach off from the site, leaving B only. Now B is the only single B on the lesion, is a good attachment point for C. So you can see B is recruiting C there. B is calling C. Specifically it is calling C to. So B and C at the point of lesion or at the point of dimer, they are there now. So UVRB and UVRC at that point, they act as a endonuclease. What is an endonuclease? Function of endonuclease: UVRB cuts phosphodiester bond of the DNA 5 nucleotide away from the lesion. UVRC, it cuts the phosphodiester 8 nucleotide away from the lesion. That is how they both function. Recall, remember, and think once again. UVRC, it cuts the phosphodiester bond 8 nucleotides away from the lesion. UVRB, it cuts the phosphodiester bond 5 nucleotide away from the lesion, giving rise to a 12 to 13 MER. This generates a 12 to 13 MER nucleotide along the long strand of DNA, and then you can see this UVRB detaches from the lesion. UVRC is remaining. Okay, UVRC, it remains. UVR, this D is then recruited on. UVRC recruits UVRD. UVRD is the helicase. UVRB and C both are removed basically. UVRD comes. UVRD is a molecule which displaces the 12 to 13 MER, that it is removes the 12 to 13 MER from the DNA, and after this both UVRD and C, they remove from the DNA. With this, now the damaged strand is removed, but as you can see, there is a gap remaining. There is a gap remaining, and this gap also has a free 3-hydroxyl end at the top DNA strand. This 3-hydroxyl end can act as a point of addition of new nucleotides by the DNA polymerase 1. DNA polymerase 1 binds to the DNA. DNA polymerase 1 refills the gap with the correct nucleotides. DNA ligase, it seals the nick. This is the whole process of how the nucleotide excision repair, it functions. So if you want to summarize this whole process in a jiffy, in 3 to 4 points, you can say nucleotide excision repair is defined as a process which removes the damaged base along with a stretch of healthy strand, and it repairs the whole DNA. This is done by a enzyme complex which is called as Uvr Abc endonuclease. Uvr A, B, they recognize the lesion. Uvr B and C, they cut the damaged DNA strand. Uvr D, it removes the strand. DNA polymerase 1 fills the gap, and DNA ligase, it seals the gap. This is a simple and very interesting mechanism of nucleotide excision repair. Now there is another mechanism which is called as base excision repair. So come, let's see its animation version first and then the PowerPoint version for your better understanding. Let's move on to base excision repair now. Come now, let's take a look at the DNA repair mechanism by a function called as base excision repair. There are various types of repair mechanisms available in the eukaryotic and the prokaryotic system, as I have already mentioned. So this is one of the next type of repair systems called as the base excision repair. Now this base excision repair system which is there, this also, just like other mechanisms, uses certain proteins and enzyme molecules which form the backbone of this repair system. So in this, some damaged DNA bases are repaired by excision, that is, it excises off certain bonds, as we will see. So this repair system works differently from the nucleotide excision repair. So if you see the damaged base which is there, that is attacked and that will be removed. If you recall the nucleotide excision system, which clips out a certain section of a DNA. Okay, as you can see here, nucleotide excision repair, it functions by clipping of 12 to 13 bases, means 12 to 13 MER of the DNA is removed and it is replaced with the correct system. Whereas base excision repair, if you see, on the other hand, it works on repairing on the defective DNA directly, means it does not harm the healthy bases as we saw in the nucleotide excision repair. So this is the difference between nucleotide excision repair and base excision repair. So in this type of mechanism, you can see certain bases which they are not supposed to be in the DNA. Okay, there are certain bases which should have not be, these are not the correct Watson and Crick type of pairing. For example, here let us say hypothetically we are seeing cytosine getting converted into uracil. Okay, if a base cytosine spontaneously deaminates, okay, so you can see here, if this is cytosine and it deaminates, it will form uracil, and uracil can be incorporated erroneously into the strand during DNA synthesis. So this is cytosine and the formation of uracil from cytosine. So if uracil forms in the place of cytosine, it would have a mutagenic effect. Okay, it will have a mutagenic effect if it is formed in the place of cytosine. That is, formation of uracil in the form of cytosine gives rise to a mutagenic effect. How? You see, normally guanine it pairs with cytosine and adenine it pairs with uracil. During DNA synthesis, DNA polymerase would incorporate these two bases in the growing strand. Means adenine and uracil, this is an incorrect nucleotide which will be incorporated in the growing strand. So as this happens during course of replication, if you see, not only this much, okay, we have seen that adenine and uracil, which is abnormal base pairing, gets incorporated into the growing nucleotide new strand formation during replication. Apart from this, few other types of damage are also repaired by the base excision repair. Which are those damage? You can see, for example, when adenine here in this figure, you can see adenine, this adenine, if it becomes deaminated, this adenine, if it becomes deaminated, then it forms an unnatural base called as hypoxanthine, and a mutagen can also damage adenine by alkylating it and converting it into 3-methyladenine. 3-methyladenine. Okay, now this adenine which is there, normally adenine which is there, it forms a base pair with thymine. Okay, this forms a base pair with thymine. But hypoxanthine, if you see this hypoxanthine, it forms a base pair with, let me just rewind you, there is a technical problem. I will just try to rewind it and show it to you once again. Let's go back. Yes, here. So if you see, normally adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it pairs with cytosine. Now 3-methyladenine is even more toxic. When it is encountered in the replication fork, it halts the DNA replication and consequently a repair system is needed. So these are the three probabilities apart from adenine and uracil abnormal base pairing. So adenine and the hypoxanthine which is there, which is pairing with cytosine. Adenine forms a base pair with thymine, this is normal. Hypoxanthine forming a base pair with cytosine, abnormal. And the 3-methyladenine forming a base pair with, it is an alkylating agent, and it keeps on changing bases. It halts the replication fork, means a apurinic or a pyrimidinic site can be formed. Consequently, this repair mutation is required, system is required, and this repair system is very vital for the survival of that organism. Base excision repair employs a variety of glycosylase enzymes which can recognize and remove the damaged bases from the phosphodiester backbone. So you can see here hypoxanthine DNA glycosylase is one, 3-methyladenine DNA glycosylase is another, and uracil DNA glycosylase is another. So this recognizes hypoxanthine, 3-methyladenine and uracil respectively when present in the DNA. The glycosylase then cleaves the bond connecting the damaged base to the deoxyribose in the phosphodiester backbone. Result is an intact phosphodiester backbone missing a base. So this is called as a AP site. It could be a lack of purine or a lack of pyrimidine. It would be a purinic or a pyrimidinic site. In general, it is called as a AP site. The next step in this base excision repair involves AP endonucleases, which specifically cleaves the phosphodiester backbone at AP sites. As you can see, the phosphodiester bond is broken and 3' hydroxyl end is left free. This 3' hydroxyl end is a very good site for addition of new nucleotides by the polymerase enzyme. DNA polymerase 1 also has a 5' to 3' exonuclease activity. As you just saw, the 5' to 3' exonuclease activity removed the bases. Now finally ligase comes and seals the nick and the repair process here is completed. The very interesting part here is of, I will just rewind you to show here the part of this DNA polymerase 1 enzyme system. So if you see DNA polymerase 1 here, it starts adding the nucleotides. At the same time, this polymerase uses its 5' to 3' exonuclease activity to cleave the strands downstream of the AP site, and then simultaneously it goes on filling. Then DNA ligase, it comes and seals the remaining nick, and the repair process is here said to be complete. So this is the process of BER. So we just had an animated look of how BER functions, or base excision repair functions. So let's get into more details about it. This is, as you can see, again another type of repair mechanism which involves number of enzymes. So base excision repair, as you can see on the slide here, it removes abnormal bases. If you saw the nucleotide excision repair, it removed the pyrimidine dimers, whereas base excision repair, it removes the abnormal bases which are formed due to uracil, hypoxanthine, xanthine, or alkylated bases. In some other organisms it may be pyrimidine dimers, etc. So these things we saw in the types of mutations and what the mutagenic agents are capable of doing. So even in the spontaneous or induced mutation, whenever this is encountered, the base excision repair may function. So this involves certain enzymes, DNA glycosylases. Now DNA glycosylases, there will be nucleotide specific glycosylases like uracil glycosylases, thymine glycosylases, so on and so forth, depending upon which nucleotide it is removing. AP endonucleases, this is the one which will literally remove the base from its point. DNA polymerase 1 for filling the gap, and ligase for sealing the gap, which you all know. So there is a removal of a phosphodiester bond on the DNA, and ligase function is finally, after the repair is over, to introduce that phosphodiester bond to seal the gap. So again, as usual, as our ritual, you should know when you study base excision repair, what is a base excision repair, how will you define it, who discovered base excision repair, because who discovered, why I am telling you, he got a Nobel Prize for that. Then DNA lesions which are rectified by base excision repair, how that mechanism works, which are the enzymes involved in this mechanism, and at the molecular level, at this level, try to understand what is the difference between studying DNA damage and repair at the intermediate level, that is 12th standard, and at graduation level is the molecular mechanism. You should be able to put forth your ideas at the molecular level, at cytoplasm level, means at the nucleotide pool level, what is happening there on the DNA, with the DNA, by which enzymes, and what is the significance of studying all this, you should be able to tell. So come join me to understand what is base excision repair. This is a type of DNA repair where you see the phosphodiester bond, that is the backbone, is intact and only the base is removed. The base is removed by a mechanism called as pinch, push and pull. Pinch, push and pull, and it removes only one single base. It is present in prokaryotes and eukaryotes. The damaged or the abnormal bases, that is the unnatural bases, they are first cleaved if you just remove one bond, that is the N-glycosyl bond. N-glycosyl bond, if it is removed, you can see here the bond which connects the nitrogen base to the sugar at the first carbon. That base connecting the sugar to the first carbon is N-glycosyl linkage. If that is removed, then the base will be flipped out of the system, out of the DNA strand. So if it is a damaged or unnatural base, then there are enzymes to recognize this. Normal bases won't be recognized, and this is removed. So the mechanism is as simple. If there is uracil, which is a abnormal base, you can call it as a damaged base. Now this N-glycosyl bond which is there, and these are the hydrogen bonds, these are the hydrogen bonds, this is the phosphodiester backbone, and this is the N-glycosidyl bond. So if this bond is broken, hydrogen bonds, they are weak bonds, so naturally this base will be flipped off. That is what the mechanism is all about. So you can see a damaged or abnormal base is specifically cleaved off from the duplex. If this is abnormal base, the N-glycosyl bond is cut and this one base is pinch, push and pulled and thrown away in the nucleotide pool. So this base excision repair was discovered by Thomas Lindahl in 2015. He got a Nobel Prize for this. What Thomas Lindahl observed was that there were many uracil molecules in the DNA. He found uracil molecules are frequently present in the DNA, which is totally abnormal, okay, for a new genetic material to function. If the enzymes for transcription, translation, DNA replication, they have to function, and if a DNA is containing lots and lots of uracil in its original wild type, native type, then it's very abnormal. So he thought that there must be some enzymatic method to handle this, and Lindahl, he isolated uracil DNA glycosylase. For the first time he isolated an enzyme called as uracil DNA glycosylase, which was having the capacity to introduce nicks or cutting the N-glycosyl bonds. So he later on detailed the molecular mechanism of BER, which is discovered, and he got a Nobel Prize for this. So the mechanism is somewhat like this. It follows a single base change recognition, it rectifies it. DNA lesions, they are rectified by base excision repair mechanisms, where many single base changes can be removed from it. Deaminated bases and alkylated bases are very easily recognized. Means these are the most common types of error repair mechanisms whenever there is a abnormality in the DNA. So BER or base excision repair can recognize very easily, or the repair of deaminated bases and alkylated bases are very common. So examples of deaminated bases are adenine and hypoxanthine, as you can see here. Adenine and hypoxanthine, this is the abnormal base. Guanine and xanthine, this is the abnormal base. Cytosine and uracil, this is the abnormal base which will be removed by BER. So this can become a very good question for your MCQs. You should have a brain for that. Now alkylated bases are 3-methyl adenine, 7-methyl guanosine. When there is a addition of alkali, then this methyl group, that is the DNA methylase system doesn't methylate this, and there is abnormality in the base. So oxides of bases like 8-oxoguanine also is a abnormal base which can be repaired by base excision repair. So there are certain enzymes involved in all the types of repair system. BER is no exception. Enzymes involved in BER, the prime enzyme, the hero for this repair system is DNA glycosylase. It cleaves the N-glycosidyl bond to remove the damaged base and flips off the damaged base away from the strand into the nucleotide pool. There are specific DNA glycosylases for each type of the damage. Means uracil glycosylase, adenine glycosylase, thymine glycosylase may be there in different types of organisms. So uracil DNA glycosylase is very common because that is the abnormal base there. Adenine, thymine, guanine, cytosines may not be abnormal, as is uracil in a DNA. So uracil DNA glycosylase has found major significance in repair mechanisms by base excision repair system. So AP endonuclease is another enzyme which has the capacity to cleave the phosphodiester linkage at the 5' end of the backbone. Now this is needed for removing the apurinic or the apyrimidinic site. What is the difference now? You see, compare this with nucleotide excision repair or the photolysis system. In nucleotide excision repair, long stretch of DNA was removed. In the photolysis system, the dimers were removed there itself, no excision. Whereas in base excision repair, only one base which is abnormal, means it is not a dimer, it is a non-bulky lesion or a small lesion. This is called as non-bulky lesion. Only one abnormal base has to be removed, and at that point a fresh base, a correct base has to be added, which will be done by polymerase 1 in prokaryotes and which will be done by DNA polymerase B in eukaryotes. DNA ligase in the end will seal the gap. So this DNA glycosylases, or the uracil DNA glycosylase, which was discovered and got a Nobel Prize. Let us see some features for that. Uracil DNA glycosylase, it flips the uracil residue out of the duplex. That is what I said, flipping by pinch, push and pull mechanism. It's simple. The cutting is called as, the recognition is called as pinch, cutting is called as push, and removing and flipping it off is called as pull. So in 3-step process, this uracil DNA glycosylase, it functions, and DNA glycosylases are responsible for the initial recognition of the lesion. Then they flip the damaged base out of the double helix and they cleave the N-glycosidic bond out of the damaged base. Now what does this result? This results into a AP site. AP site can be a purinic site or a pyrimidinic site. Okay, so there are 2 categories of glycosylases. Now this is very important. If you see, there are 5 motifs. This 5 colors, they show the 5 motifs of the DNA glycosylase, uracil glycosylase, which has a developed active site inside it. It recognizes the wrong base, pinches and pulls and throws it away. So there are 2 categories of this enzyme. One is monofunctional, other is bifunctional. Now bifunctionals are very effective. How that? Monofunctional have only glycosylase activity, whereas bifunctional, it possesses the AP apurimidic site lyase also. In monofunctional type, it only recognizes and the N-glycoside bond is cut. The remaining part has to be done by AP endonuclease enzyme. That is the apurinic or apurimidic site endonuclease enzyme. But in a bifunctional type of uracil DNA glycosylase, listen carefully, in the bifunctional type, it can recognize the wrong base, it can cut, it can remove, and it can also, what you can say, do the lyase, AP lyase activity also. So this is, of course, filling has to be done by polymerase. Therefore, bifunctional glycosylases, they can convert a base lesion into a single stranded break without the necessity of AP endonucleases. Glycosidic bonds, they follow a pinch, push and pull mechanism using the 5 motifs. Now these are the active sites of the protein or the enzyme molecule. Now the steps, these are very simple, as it involves only one base. Okay, one abnormal base is recognized, that is uracil DNA glycosylase. It recognizes the uracil, let us say, uracil glycosylase, it cleaves the N-glycosidic bond. This results into a AP endonuclease, AP site, which is then flipped off, that is by the enzyme endonuclease. AP endonuclease cleaves the phosphodiester backbone. Of course, uracil glycosylase flips off the base, therefore there is a result of AP site. Now this is removed by 2 mechanisms. One is resulting into a long patch, other is by a short patch. This glycosylase enzyme can function by just removing the only one single base and filling, or just like the nucleotide exchange, there can be a long patch of some 4 to 8 or whatever, depends upon different species. A long patch can be removed. So DNA polymerase 1 or the beta, it fills the gap, ligase, it seals the gap, and the damage is repaired. So this is how the BER functions. It has certain significance in the survival of a cell or organism. BER is most important for removing the bases which are altered, rather than a wrong pairing. The altered bases which change its chemical structure and still have the capacity to wrongly form bonds with normal pairs, that are removed. DNA bases or the damaged bases, they can create mutations by mispairing, and this can halt the DNA polymerase enzyme, stopping it from replication. Many effects are there. So this base excision repair can remove uracil, xanthine, hypoxanthine, oxidized bases, alkylated bases, and mutations in the BR machinery, which can lead to cancer. So this is all about the base excision repair. Now the final and the last type of excision repair is the mismatch repair system. So I hope you understood the base excision repair. Repeat your visuals once again. If you have any difficulty in understanding, go through this presentation a couple of times. It's very easy to understand. Once you understand the molecular mechanism and the principle of this, all things will automatically fall in your place in the brain. Okay, so now let's move on to understand what is mismatch repair system, the final of the repair system for your curriculum or the syllabus, mismatch repair system. So friends, that's all about NER and BER. In my next lecture, I'll be talking about the final type of repair system. Again, a complex assembly of enzyme which takes place, a very interesting assembly of the proteins on the damaged DNA and how it repairs the cell's DNA so that the cell can function normal in its metabolism, which is called as a mismatch repair system in my next video. Till then, goodbye.